we have two toxins. One is exotoxin and the other is endotoxin. Endotoxins are the parts of the cell wall. So they have two jobs. One job is structural unit and during the life of bacterium, and the second job is showing toxicity after bacterium going to die by breaking the cell wall and release these endotoxins. But regarding to the exotoxins, they are produced during the life of bacterium, excrete outside for a specific tissue, a specific target place. So endotoxins don't have specific target plates. They can mostly be effective on different cells, different tissues, different organs. But exotoxins, mostly which they are produced according to the bacterial producer, they are effective to special parts, special target tissues. Then we compared them in different things, in different items. Then we showed you the chemical structure of endotoxin. So it is a part of cell wall, lipase and core oligosaccharides and O antigens. But the main and important part is lipid A. All parts are necessary for toxicity, but main part is lipid A. Again, we showed you the appearance of a gram-negative cells. Lipid A consists of fatty acids and phosphorylated glucosamine disaccharides. It is lipid A. Toxicity is related to this part. And another parts are core polysaccharide and repeating units or O antigen. In total, we call this figure as lipopolysaccharide, LPS. So this lipopolysaccharide, lipid A, core, and O antigen in total is an endotoxin. And again, remind you, lipid A is responsible for toxicity of endotoxin. The main part is lipid A. LPS reaction, I think I explained that. What is the main aim and purpose of our body in front of bacterial pathogens? Our immune system, specific and non-specific, white blood cells, macrophages, antibodies, and whole body wants to kill the pathogen. As well as we use antibiotic to help our immune system to kill the pathogens. But when we kill them, it is not the end of the story. When they are killed, the pathogens when are killed, now it is starting to release LPS or endotoxins. So in a sense die of reaction. Die of reaction means you are killing pathogenic organisms. So it should be done. It is important to kill them, which is good thing. Yes, it is good. It has to be managed through though as there shouldn't be a reason to suffer through it, but problem not to be finished. Some problems finished, some new problems are started after the death of the pathogens. The body can handle reasonable of die off pretty well. When this becomes too much, when there are too much pathogens and too much of the pathogens are killed, inflammation will increase dramatically. Inflammation increase dramatically. Blood pressure drops too much. Low blood pressure and less oxygen is transported to tissues and organs. So many problems are started by killing gram-negative cells. Excess endotoxin can also damage DNA. So it is another problem. So when LPS are released in the body, when the endotoxins are released too much in the body, according this die of reactions, what can we feel? We feel fever, high white blood cell count, low iron, blood clotting disorders, thrombocytopenia, platelet aggregation, constipation, headache, muscle pain. And LPS also produces considerable amounts of ROS. What is ROS? Reactive oxygen species and other free radicals. So they are not good. Free radicals, they are effective on different parts of the cells. So what should we do? Should we kill them or we shouldn't? Yes, we should kill them. So this problems continues. What should we do regarding to the fever, blood cell count, low iron, and these things? For minimizing the die off reactions, we should understand how to manage this die off. So when one person is in the hospital with heavy infection with a gram-negative and physicians are using antibiotics, the patient should do something more. Like what? Drink plenty of water. So you know, many of the lipopolysaccharides can be filtered by kidney if you have sufficient water drinking. So drink plenty of water as much as possible. Consume more fats. So fats come around the LPS and make it neutral. Follow a mainly paleotype diet. What is paleotype diet? Typically include lean meats, fish, fruits, vegetables, nuts, and seeds. What are we doing? We try to minimize and neutralize the effect of LPS. Quercetin, it is a plant pigment, it is a flavonoid, and it is found in many plants and foods such as onions, green tea, apples, berries. EPA and DHA both are omega-3, and they are abbreviation of these names. ALA is found mainly in plant oil such as flaxseed and soybean and canola oil. DHA and EPA are found in fish and other seafood. And it is important to have exercise. So by exercise, you make your metabolism maximum, filtration by kidney maximum, and you try to make the LPS in minimum amount. Another thing sometimes may be happening is regarding those people which many times during the year and in continuous years several times have heavy infection with gram-negative bacteria. So some people in different cases have many problems. They have many problems with gram-negative bacteria. And accordingly, going to physician, use antibiotic, and if they are alive, so they could kill them. But when they kill the gram-negative bacteria, there are a plenty of endotoxins are produced in their bodies. LPS can cause leak gut, so bad effect on intestine, and intestine is leaky. If the immune system is constantly being activated to deal with endotoxin, it can eventually lead to the development of autoimmune disease. Autoimmune disease. So when for many times, several times, for continuously, one person have infection with gram-negative, and immune system all the times are active against gram-negative endotoxins, after some time there is autoimmune disease. It means immune system attack to the host cells, to the cells of the person. Immune system attack to the different cells of the patient body. And maybe this autoimmune response, this autoimmune disease can be responsible for diabetes type 1, rheumatoid arthritis, thyroid disease like Graves, Hashimoto disease, IBD, irritable bowel disease, MS, multiple sclerosis, and other disease. So the first disease is related to the gram-negative bacteria. That LPS are continuous production of LPS in body of one person may be causative for secondary problems, secondary disease, secondary involvements like diabetes, rheumatoid, and arthritis, thyroid disease, and so on. So first, we should try not to be infected. So some people which they live properly, they have less infection during the life. But some people with bad behavior, bad lifestyle, mostly in every year they have several times infected with gram-negative bacteria. So maybe it is not restricted only to the pathogen, and it comes to the another disease and another problems, and sometimes maybe it can be life-threatening. Okay, so right now, approximately we explained some pages, page regarding endotoxin, and from page 13 till end of the semester, we want to focus on exotoxins. So endotoxin, we had only one. So the endotoxin of all gram-negative bacteria were the same in structure, behavior, and all the things are similar. Endotoxin of gram-negative bacteria. That exotoxins have many variations, many differences. So one exotoxin from one microbe may completely different with another exotoxin. And sometimes one bacterium produces many different exotoxins, which each of them do specific jobs. If you remember, unlike endotoxins, exotoxins are produced during the life of the bacterium. It is secreted outside the bacterium and show its activity. Unlike the endotoxin, which it was lipopolysaccharide, it is protein. Unlike endotoxin which was resistant to heat, it is heat sensitive proteins. Heat labile proteins secreted extracellularly by certain species of bacteria, which diffuse into the surrounding medium. If they are in the liquid medium, or they are in solid medium, or they are inside our body, they can produce their exotoxins. And these exotoxins are one of the virulence factor of the pathogen. It is one part of the power of a pathogen which can produce exotoxin. It is soluble in water, so hydrophilic. Exotoxins are hydrophilic and very easily can move, can diffuse from one part to another part in liquid or solid media. These are highly potent, means they are highly powerful. And if only a little of them can be lethal, can kill any living things. they are highly potent in minute amounts, very small part, and include some of the most poisonous substances known. So we have many toxins, we have many poisons. Some are related to living things and some synthesized, some come from environment, but toxins from bacteria are very powerful. For example, if we have only 3 kilograms of botulinum toxin, you know, botulinum toxin is a toxin produced by Clostridium botulinum. It is not produced inside body. It is produced in food, like canned food. Sometimes we buy canned food from outside, from shops, and they receive it from factory. So some canned food are produced in factory, and some canned food are homemade and produced at home. And the door is completely closed and sealed, and mostly it is without oxygen. So this anaerobic bacterium, when grow inside canned food, produce toxin. And if we eat an infected canned food containing toxin of botulinum, we go through the disease of botulism. The disease of botulism. If we have 3 kilograms of botulinum toxin, it can kill the entire human population of the world. So only 3 kilograms of botulinum toxin can kill all people which now they are alive in the world. If we have another toxin, for example, tetanus toxin. Tetanus is another toxin which is another exotoxin which is produced by Clostridium tetani. Okay. So if we have only 1 milligram of the toxin of tetanus toxin, it is sufficient enough to kill a million animals like pigs. Yes. So when we have pathogen, why did we study them? Because when we know clearly our enemies and our friends, we can use them. For example, when you know very bacterium, you can make toxin. If you know very well the toxin of bacteria, you can produce toxin. Toxin is the modified toxin by formulating heat or another ways which doesn't have toxicity and have some parts of toxin. And if you use it in a person, can produce antibody which is on original toxin. So you can immunize yourself by toxin against exotoxins. And for example, Clostridium botulism, which I read that 3 kilograms of toxin is sufficient to kill all people. Now in biotechnology, this toxin, very small amount of this toxin, sometimes using by physician for what? For example, there are some folds on face. So by botulism toxin removing these folds and show the person younger than he is. Yes. So first thing is that they are pathogen. First thing is that how they have pathogenicity, what are their toxins, how can we defend us. It is main subject. And second subject, they have toxin. Can we use them? Can we use those for medical purposes? Yes, of course we can. Like what you wrote. Okay? So exotoxins are generally produced by gram-positive organisms like Corynebacterium diphtheria, Clostridia, Streptococcus hemolyticus, Staphylococcus aureus, and Bacillus anthracis. Certain gram-negative bacteria like Escherichia coli, like Shigella dysentery, Vibrio cholera, Aeromonas produce exotoxins. So, some gram-positives and some gram-negatives can produce exotoxins, but all gram-negative cells can produce endotoxins. Endotoxins, all gram-negative. Exotoxins, some gram-positive, some gram-negative. So later on, I write who are those microbes which can produce exotoxins, and one by one we explain what are their mechanisms of action. What are their mechanisms of action? They spread via bloodstream or sometimes through nerves. So when one toxin can reach to blood or nervous system and spread in the body, it is not good. Sometimes some infections are related to a specific part, a specific tissue. So they are local infections. But sometimes some infections can spread all over the body. So they are more dangerous. Mostly they use blood and nervous system to going from different parts of the body. They are highly antigenic. So highly antigenic, it's okay because they can induce immune system response. So when they are antigenic, immune system know them and reflect and can be converted into non-toxic toxoid. Yes, we can do it. How? By addition of formalin or acid or heating. So if we have some toxins, exotoxins, we can add formalin, it is a chemical liquid, or acid, or by heating, and have some modification on toxin and change it to non-toxic toxoid. Sometimes we want to try to stop a pathogen before coming to body. For example, you use a vaccine because you want, if after some time the wild type pathogen come to your body, your immune system to be ready. So you use vaccine. But sometimes you want your immune system to be ready against toxins. So instead of whole microorganisms attenuated, weak, or killed, instead of organism, first you receive toxin, you have some modification and change it to toxoid form. Toxoid form means it has many similarity to toxin, but without toxicity, without virulence. So what happened? So this antigen, toxoid antigens, activate, induce immune system, antibodies are produced, and it is not toxic, so it doesn't have any problem to the body. But if in the future the original bacterium produce toxins, your immune system is ready to suppress it. Toxoids or antitoxins are non-toxic, so it is good. If they were toxic, it was not good. Antitoxin or toxoids are non-toxic but antigenic, so they are not toxic, so it is good. And antigenic, so they can induce immune system and activate immune system, so it is good. Both are good. Toxoids are non-toxic but antigenic. Retain the ability to produce antibodies. Okay. What is this figure related to? This figure wants to explain some bacteria far from the target cell produce toxin, and the target cell absorb it, like type 1 and 2 secretion system. But some microbes, some bacteria go very close to the target cell, going very close to the target cell, attach it and inject, yes, like a syringe injection to one person. Yes, you have one medicine, so you need to have injection by syringe, and syringe has a needle. So bacteria go and have a contact and inject toxin inside the target cell. In figure wants to explain gram-negative bacterial protein secretion system, and it wants to say in general it is two types. In the type 1-2 secretion system, bacteria secrete toxins in the extracellular space, toxin excreted in the extracellular space in the upper image. In the type 3-4, in the type 3-4 secretion system, bacteria directly secreted toxins into the cytosol of target eukaryotic cells through the secretion apparatus, by their secretion apparatus. But what is the secretion apparatus? how they can inject. Do they have a syringe? Do they have a needle? Here you can see this. This is not pili. This is not flagellum. This is a needle component. This is a needle component which is present in some gram-negative bacteria like Pseudomonas aeruginosa. It is type 3 secretory apparatus. Type 3, yes. It is type 3 secretory apparatus. The needle, where is the needle? So this is inner membrane. This is periplasmic space between inner membrane and outer membrane, so it is gram-negative. It has a basal component. Basal component, it has like pili, like flagellum, but it is not for motility. It is not for attachment to bacteria to each other. It is working for attachment and sending excretion. By what? By this needle component. And at the head, it has a cap protein, and here is hollow. Exotoxin coming from this hollow place, coming, and then by this directly inserted to the cytoplasm, of target cell. Target cell is human cell. Human cell is a eukaryotic cell. Pseudomonas aeruginosa type 3 secretory apparatus, the needle, complex, or injectosome. We call it injectosome. What is this for? Previously, you understood there are some pili, by pili bacteria attached to each other, by pili bacteria attached to, or fimbria attached to the target cell, but it is used for attachment and injection of toxins. So, two jobs: attachment and injection of exotoxins. What do we call it? We call it as injectosome. It is injectosome. The type 3 secretory apparatus comprises many protein components. You can see by blue color. It has different parts, like different parts of pili, like different parts of flagellum. So it is injectosome for transferring directly an exotoxin inside the cytoplasm of human cells. Okay, what do we want to explain here? We want to say, we want to talk about types of exotoxins. Exotoxins can be classified in three groups. Group 1 means the toxins which have two subunits, like this. It is an exotoxin. It has A subunit and B subunit. It is an exotoxin with two subunits. One is A and second is B. The other exotoxin we call as super antigen. The other is super antigen. And the last one is membrane disrupting exotoxins. Membrane disrupting exotoxins. So AB exotoxin, superantigen exotoxin, membrane disrupting exotoxins. So a few minutes we want to discuss what are they? What are they and what are their action? First, AB toxin or AB exotoxin. As you see, what can you see? They have two components, A and B, yes. A subunit, B subunit, yes, we can see. B component binds to a specific cell receptor, so B is used for attachment to the target cell. It can understand where is the target cell and where is not the target cell. So B subunit find the way. B subunit find the target and complete toxin attached to target by B subunit. After attachment, B subunit facilitate the internalization of A. So B subunit attach and help A subunit as well come inside the cell as an endosome. Then there is a dissociation between A and B, and A come to the cytoplasm, and A is the only responsible for problems. So A and B come together to the cell, and B show the way, then they dissociate, and only A is working inside the cell and make it in problem. Here again you can see it is AB toxin. This red is A and this blue is B. How B understand what is the target? On any cell there are many. So those cells have receptor matched to the anti-receptor on B can cause matching. So subunit of B trying to find its own receptor. And any cell have this receptor is the target cell. So now A is attached to B, B is attached to receptor, receptor is on the target cell. After successfully attachment of B as well as A to receptor, endocytosis is started. Endocytosis. And an endosome is produced containing B and A and receptor. This is a vesicle. This is a vesicle produced inside the cytoplasm. Then there is dissociation between A and B. Dissociation between A and B. A coming out to the cytoplast and start virulence, start problems, that B subunit accompanying with vesicle rejected outside by the cell. So B rejected outside, that A only can remain inside the cytoplast and cause the problem, cause the toxicity. Okay? So B is responsible for attachment, and after going inside, then rejected, and only A remaining in the cytoplast for further problems. What was this? It was related to whole bacteria which produce exotoxins, which their exotoxins contain two subunits of A and B. What is their target cells? Target cells is related which anti-receptor the toxin has and with which receptor on which cell can be matched. So accordingly we understand why some exotoxins can invade nervous system, why some can only attack to the intestinal cells, why are they so specific. It is not like key and lock. Yes, you have a key. You can only open the door of your home. Why? Because your key is related to the lock of your home. It is not related to the another home. You have one key, you can open the door of your car. Your key is specific to the lock of your car. So the key is receptor and the lock is the B subunit, and they should be matched. If not matching, no penetration and no toxicity happening to the cell. If matching, story starts. The story can be starts. What can we see here? Here we want to show you some bacteria which contain AB toxins. We want to tell the names of those bacteria which they contain exotoxins having A and B subunit. Like what? Some neurotoxins. Some neurotoxins are available. What does it mean? They can attack to nervous system, to neurons. Like what? Like Clostridium botulinum. Like Clostridium botulinum, which cause botulism. What is the name of toxin? Botulinum toxin. Clostridium tetani, another, with AB toxins, with completely opposite direction, toxin is work. This is tetanus, and the toxin is tetanospasmin. Enterotoxins, those exotoxins which they are effective on our intestinal cells. Enterotoxins, like enterotoxigenic E. coli have several different groups. One group of E. coli is related to the enterotoxigenic. The disease name is traveler's diarrhea, and the name of toxin is heat labile enterotoxin or cholera-like toxin. The other toxin which is effective on intestine, it is Vibrio cholera is the causative agent. Vibrio cholera. The disease we call as cholera, and the toxin which is produced is cholera toxin. Some toxins are effective on the cells, on some cells. We say cytotoxins. Like what? Like bacillus anthracis. The disease is anthrax, and we call it edema factor or lethal factor. Don't worry, because one by one it is we are talking which items we want to explain afterwards. So first we go to the class with the polio, then talking about the then enterotoxin E. coli, then the cholera, then bacillus anthracis, and we explain these items afterwards. Yes. Number one was a toxin. Yes, it was number one. What was number two? Super antigen exotoxins. Another exotoxin is super antigen exotoxin. What is this? What is the super antigen exotoxin? Super antigens are carried over the bacterial proteins that bind simultaneously means at the same time to the domain of T cell receptor. So connecting T cell receptor and alpha chain of the 2-MHC molecule. In normal case, when there is an antigen first connect to antigen presenting cell, like many white blood cells or microphages, and this antigen presenting cell have antigen and go into the T cell. Together, and there is an attachment between MHC class 2 of antigen presenting cell and T cell receptor of T lymphocyte, and T cells induce antibody production, but it is for normal antigens. Exotoxins are not normal. Exotoxins, some of exotoxins are not look like a simple antigen. We call them super antigen. They are not simple antigen. They are super antigen. So what? Without connecting antigen presenting cell or APC to TCR without directly, it can indirectly connect T lymphocyte 2 antigen presenting cell. So some exotoxins are not so simple antigen. Because they are not simple, we call them super antigen. And instead of connect these receptors to each other, they can connect these two cells together directly without going to the receptors. Super antigens cause cross-linking of T cell receptors, TCR, it is cross TCR and MHC class 2, leading to what? Leading to uncontrolled activation of T cells. So these T cells going and activated and working uncontrolled. Whatever is working uncontrolled is not good. So it is not good. Some toxins activate T lymphocytes and it is highly working continuously. So this is T cell, this is antigen presenting cell, this is T cell receptors, this is MHC2 molecules. So we told you that super antigen, which come as an exotoxin of some bacteria, connect to V beta of T cell receptors of T cell and alpha 1 of MHC2 molecule of antigen presenting cell and activate T cell so much. Activate it so much. The binding of T cell receptor, TCR, T cell receptor, this part, with major histocompatibility complex 2, major MHC, major histocompatibility complex 2, MHC, it is what? It is this, MHC2, which can ultimately result in life-threatening autoimmune response. So when this happens, this super antigen is responsible for highly too much working of T cells with too much production of antibodies and too much activity. So this much of activity is not good. This much of activity is not good. And it goes through the autoimmune response. Means immune system attack to host cells. Autoimmune system attack to the human cells. So it is not good. So super antigens bind simultaneously to V beta domain of T cell receptor and to alpha chain of class 2 MHC molecule. This cross-linking of T cell receptor and class 2 MHC molecule produces an activation signal that includes T cell activation and proliferation. We have too much activation of T cell, we have too much proliferation and too much number of T cells. The number of T cells sharing V beta domain is high as a consequence of superantigen activates large number of T cells with different specificity. So what? The massive activation of T cells results in overproduction of T helper cell cytokines, tumor necrotizing factor, alpha, interleukin 1, interleukin 6, and these items leading to systemic toxicity, multi-organ failure, and even death in some cases. So there is a systemic, so means whole body has toxicity, receive toxicity, and many organs cannot work properly, and sometimes it leads to the death of the person. So when we say super antigen activate T cells, we shouldn't think it is good. So make it crazy. Super antigen make our immune system crazy, and instead of defend us, attack to our body, attack to our body. The job of immune system is defend and attack to the bacteria, defend from us and attack to the bacteria. My super antigen immune system is crazy, and it is friend of and hydrogen and immune system are friend to attack to our body. The last group is membrane disrupting toxins. Membrane disrupting toxins. Membrane disrupting toxins lyse host cells by disrupting plasma membrane. So membrane disrupting, its name shows their activity. Excuse me 1 second, I have emergency. Excuse me. So its name shows its activity, membrane disrupting toxins. So those bacteria with these types of toxins attack to the cytoplasmic membrane. So cytoplasmic membrane is a vital part. By any disruption, it leads to the death of the cell and group of cells in a tissue or organ. Membrane disrupting toxins lyse host cells by disrupting plasma membrane. Yes. Like what? Like leukocidins. What is leukocidin? As its name shows, this toxin is effective on leukocytes. Leukocytes are white blood cells, defensive cells. So we are waiting. Phagocytic leukocytes kill the bacteria, but we see some bacteria by leukocidins kill phagocytic leukocytes. So it is not good. Some bacteria can kill some of our defensive cells. They can kill some of armies, some of our soldiers. Hemolysins. Some bacteria produce hemolysins, which they are effective on cytoplasmic membrane of erythrocytes, red blood cells. How disrupt? How they can disrupt? By forming protein channels. So many materials go inside and go outside, and the cell will be killed. Streptolysin. Streptolysin. Hemolysins produced by streptococci. Some bacteria which can produce membrane damaging toxins or cytotoxins are, for example, Clostridium perfringens. The disease is gas gangrene, and this toxin we call as alpha toxin. Staphylococcus aureus. The disease is wound and other infections. They produce leukocidin, Streptococcus pyogenes, pharyngitis. They attack to pharynx and produce pharyngitis and other infections. And they have streptolysin O. Streptolysin O. So they are three groups. AB group, super antigen, membrane disrupting toxins. Now only 2-3 minutes we explain regarding to the first. No, first we go through a table. In this table, we see what do we want to do next lectures. In next lectures, we want to talk regarding botulism. The causative agent is Clostridium botulinum, which it has AB exotoxin. Then we go through the tetanus, which is produced by Clostridium tetani. The toxin is AB subunits. Then we go through diphtheria, which is caused by Corynebacterium diphtheria. Again, it has AB subunits. Scale skin syndrome is produced by Staphylococcus aureus with AB subunits. Then we go through cholera. It's produced equilibrium cholera, A, B supplements, travelers, diarrhea, enterotoxigenic Escherichia coli and Shigella, A, B supplements, gangrene and food poisoning related to Clostridium perfringens and other Clostridium, membrane stopping, exotoxins, antibiotic-associated diarrhea, Clostridium difficile, membrane disrupting exotoxins, food poisoning by Staphylococcus aureus, superantigens, toxic shock syndrome by Staphylococcus aureus, superantigen. It is what we want to do in next lectures. Okay, tonight I send you the PDF file, and as you know, next week Iranian New Year started, and we go for a few days going to the holidays. So after holidays we can continue our lecture. So I'm so glad to finish this present year with you, and I'm happy to start new year again with you. So a few weeks, all of us, we can have some rest regarding to the study and teaching and studying, and then very freshly we will continue. If there is any question, please ask me. If there is not, we can finish this lecture right now.